It's my pleasure to now introduce Dr. So-Youn Kim, who is Assistant Professor in the Department of Obstetrics and Gynecology, the Department of Biochemistry and Molecular Biology, Cancer Research and Molecular Genetics and Cellular Biology, and a member of the Fred and Pamela Buffett Cancer Center Olson Center for Women's Health at the University of Nebraska Medical Center. Dr. Kim earned her doctorate of philosophy at UC University in Seoul, Korea, and I had the pleasure of working with Dr. Kim in 2 labs at Northwestern University, one in Dean James Larry Jameson's laboratory, and the second in Dean Teresa Woodruff's laboratory. She is now an NIH-funded researcher in Nebraska, and her lab works on elucidating the pathways of oocyte death by genotoxic agents and developing the fertoprotective agents. A recent paper published in Journal of Reproductive Immunology found evidence of inflammation in human ovaries even after several years post-cancer treatment. It is my pleasure to welcome Dr. Kim to the podium.
Hello, everyone. I am So-Youn Kim. It's really nice to see you, everyone, here today. Thank you for inviting me, Oncofertility Committee, for giving me this great opportunity. I'm truly honored to be to present my talk at Oncofertility Consortium. I have nothing to disclose. As everybody knows that pediatric cancer patient number is increasing over time. So if you look at CAC2 survival rates, it's getting increased from 70s to 21st century. So I marked to red box. If you look at here, leukemia and lymphoma and other brain cancer, everything is increasing almost twice over time. And then 400,000 pediatric cancer survivors are now in USA, and 73% will have a chronic health condition, and 42% will have a severe condition after treatment. Why it happened to kids? So most of endocrine organs are very susceptible with the chemo or radiation therapy. If you look at here, thyroid gonads are severely affected, while pancreas or other organs are not much severely affected compared to endocrine organs. So after getting the chemo or radiation therapy, they can develop a lot of disease after treatment. For example, if they can get some damage in brain, then they can develop hypothalamus pituitary endocrinopathy or precocious puberty, etc. And we are now really want to care about their next generation, also their health. So we should think about ovary and testes as well. So puberty or hormone secretion will be affected by the dose treatment. Why is it? So fertility rate will be decreasing over time. The reason is the oocyte has finite resources. When a woman is born, they have so many germ cells inside the ovary. However, after getting gonadotoxic treatment, they will lose most of follicles, and then they will become menopausal status in most of women. So they will have a menopausal status of hormone status, such as low estradiol and inhibin hormones. And also at the same time, they will increase FSH and LH high level. Because they will lose most of the ovarian follicles inside the ovary. If they're getting the chemo or radiation therapy, they will lose most of the follicles inside the ovary, and they will become, the ovary will become, they will lose most of the follicles, and they make a premature ovarian insufficiency. So as you know, there are many standard options and experimental options right now. So we can isolate, we can take out the cortical strip from the ovary, and then also we can store those the tissues into liquid nitrogen, or we can isolate the follicles from the cortical strip and then save those follicles for future use. However, there are disadvantages such as challenging in severe clinical situation and expensive and unaffordable and invasive. And it's really hard to maintain endocrinological balance for a long time. So many people, many lab, and then many people trying to figure out how to culture ovary. And then right now, it's really successful for culturing from primordial to ovulation stages in vitro. So, and if you will see here, Dr. Zhao. Dr. Zhao was successful for isolating human follicle and make mature follicles. It was many amazing technological advances in our area. I am really interested about primordial follicles. The reason is that it is still we cannot stimulate prepubertal girls with hormones. Also, it's not easy to isolate and culture single primordial follicle most of the cases. So my lab is interested in to develop certain inhibitor or some medicine to prevent primordial follicles, ovarian reserves in the body. So if the name is a photoprotective reagent. So photoprotective reagent should protect the germ cells inside the body. At the same time, they should not compromise the chemotherapeutic treatment. So eventually, the goal is to maintaining women's health in women's systemic health. So today I will talk about, first of all, it's radiation therapy, and then second is chemotherapy, and third is immunotherapy. So first of all, I wanted to introduce about radiation therapy. It has been, it's published, but we should still work on it, and it's really amazing advance in this area as well. So we have been using X-ray, and then currently people trying to move to proton therapy. The reason is that X-ray will affect will kill the tumor, but at the same time will induce a lot of damage in other organs as well. However, proton therapy will give the energy to the cancer cell and kill the cancer cell only. So decreasing side effects. So the reason is that proton therapy will give energy to the only cancer cell, and then when proton reach to the cancer cell, they will have a bragg pit, and then give a high energy to cancer cell. If you look at the right side of a graph, x-axis is the death, and then y-axis is the dose. So if you think about the left side to give energy, and then X-ray will have a high energy from the beginning, and then X-ray will gradually decrease the energy resources. And then when they reach to tumor, they will give energy. However, they also affect other organs as well. But proton therapy, they will give a low energy from when it's treated to the patient. They will go to low energy, and then when it reaches to the tumor, they will get a high peak of the energy, and then drop the energy right away, and then decreasing the damaging other organs. So we tried to prove this concept for saving ovarian follicles. And if we think about tumor next to the ovary, and then we tried to the isolating mouse, small day5 mouse ovary, and then also we tested a mouse. And we made the cassette to put mouse, and then gave X-ray versus, and then or also proton. And if you look at the right side, graph A, X-ray gives us so much energy, and then the star, meaning a star indicates the ovary. So X-ray gave a lot of energy to attack the ovary as well, and including other organs. But proton does not affect proton. We can control the distance of the energy level, so we can save the ovary. So after our experiment, we concluded that control mouse ovary contains a lot of primordial follicle and then also growing follicles inside the ovary. But after giving 1.8 grade of X-ray, it removed or wiped out all follicles inside the ovaries. However, when we gave 1.8 grade of a proton, we could save a lot of primordial follicle, including growing follicles. And however, we cannot reach it. We cannot use the prototherapy everywhere. So in USA, only 30 centers installed in among 50 states, and also 2 places are under construction. And then also we should think about leukemia patients, something like leukemia patient. We cannot use a proton therapy for those patients as well. So there are limitations, but we should think about for future using of the therapy as well. How much do we know the chemotherapy-induced ovarian damage? As recently, Dr. Zhu published a paper, and then she reported that after giving chemotherapy, and then she traced the long-term consequence, and then she collected the human and rhesus macaque and the mouse tissue, and then look at the histology, and then she found that there are so many macrophages infiltrated inside the ovary. We didn't expect the consequence of the treatment. Also, we do not know what they do inside the ovary. So we should think about how we should figure out this, and then how can we make ovary and follicle maintained inside the ovary at a healthy status. So far, there are so many photoprotective reagents that have been reported. However, we do not know what kind of photoprotective reagent we should use in clinical-wise, also in research-wise. The reason is that we still do not know how oocyte responded, ovarian follicle or oocyte responded to chemotherapeutic treatment. Some people think that follicle will be activated by chemotherapeutic treatment. However, most of people, including me, we think follicle would die by chemotherapy treatment. So currently, most promising, and then a lot of promising inhibitor were proposed dependent pathway study, molecular level study. Basically, DNA damage will induce a CHEK2 inhibitor, CHEK2 molecule, and then activate CHEK2, give phosphorylated TAP63 inside the oocyte, and will give apoptosis. Will induce apoptosis in oocyte. So a lot of paper came out from Dr. Volkan's lab. Also, Dr. Carla also published a really great paper mentioning that follicle would die by apoptotic pathway. So loss of PUMA, the apoptotic marker, protects the ovarian reserve during DNA damaging chemotherapy, and then preserve fertility. Also, really fascinating paper just came out from the Carla-Holt lab that oocyte has this ability to repair DNA double-strand breaks and to restore genetic integrity and protect offspring health. So we wanted to know we can repeat this experiment. So I will show you the data later. So I wanted to know, really oocyte would be affected by chemotherapy treatment. And also we wanted to know more than that of a primordial follicle, what's going on inside the ovary by chemotherapy treatment. So this is a primordial follicle. It's a healthy primordial follicle. You can see beautiful primordial oocyte and covered with pregranular cell. And then they will grow to secondary follicle and covered with multi-layer of granular cell. They contain a lot of healthy mitochondria. And after giving one injection of chemotherapy in cisplatin, they induce necrotic oocyte inside the primordial follicle. And there was a secondary follicle, they lost granular cell. However, in those secondary follicle oocyte are totally fine. In the mitochondria, they lost most of cristae. Why ovarian reserve sensitive to gonadotoxic reagent? The reason is that oocyte contains a P63 molecule. I wanted to emphasize of P63, how P63 molecules is really important for oocyte health, and then why we should care about P63. I will introduce in this session. So P63 will be turned on early stage of embryo day 7.5, and they will turn off when oocytes start to recombination, the chromatin recombination, and then they will turning back when oocyte is arrested, and then they maintain until early secondary follicles. P63 only expressed in ovarian follicle, not testis, and then they will be expressed only healthy follicles. If we give them radiation, then they will get damaged. They will not express of P63 molecules. P63 molecules, according to the German group, Dr. Volkan, showing a lot of data that P63 molecules are stayed as a dimer status inside the oocyte, and then they will get phosphorylated by DNA damage. They will usually give most important phosphorylation site is after exon 13, and then they will get damage of DNA, and then they will make a tetramerization. So after knowing this kind of a concept, and now I wanted to see expression of a P63. After giving cisplatin, I found that the P63 is fascinating. They would not overlapped with apoptotic markers. So BAX TAP73 and c-ABL, it's not overlapped with the P60 expression. That means that those green positive follicle would die, but red positive, P60 positive oocyte will survive. So, and then we wanted to know how much P60 is important. So we tested our cyclophosphamide, radiation, doxorubicin, cisplatin. The reason is that radiation will induce DNA damage. Cyclophosphamide will make a DNA adduct, and then doxorubicin will be a topoisomerase inhibitor, and then cisplatin will DNA adduct as a platinum-based DNA alkylating agent. They will make a DNA adduct. That means all these kind of chemo will kill the tumor cell with a different mechanism. However, we do not know their response to inside the oocyte. So we made the oocyte-specific knockout mouse model using GDF9 ICRIS system. And then we tested, we confirmed the P60 knockout, and then we tested cisplatin. We found that the condition knockout mouse after treatment of cisplatin, they protected the most of oocyte, and then we concluded that P60, if P60 is not there, oocyte has ability to survive from against the cisplatin and radiation. We also recently tested our cyclophosphamide. As we also found that the top panel is staining with the P63, so we could not see P60 staining, but bottom is DDX4 oocyte marker. We could see cyclophosphamide also induce most of the follicle death. However, P60 knockout, we could see surviving follicles. So we confirmed the counting data, and then also we confirmed the doxorubicin. So as showing that previously, and then P63 was phosphorylated by doxorubicin treatment, and we wanted to confirm the P60 is really critical for inducing oocyte death by doxorubicin, and then also we confirmed the after making a knockout, doxorubicin didn't kill the primordial follicles, and then we wanted to know how it's oocyte healthy or not. So we made a fertility cage after giving cyclophosphamide in knockout, and we could see so many pups after this P60 knockout mouse model. And then when we harvested after 5 months later, wild type with the CPA treated mouse ovary, it become really small, and then it's showing the aged style of ovary. However, P60 knockout with the cyclophosphamide, they still developing. They have a developing follicle and the corpus luteums, and then they totally fine for delivering pups. And then also we found it really interesting in primordial oocyte. Primordial oocyte will have a respond a different way by radiation and cisplatin. So we give the X-ray or cisplatin. They respond a different way to express the p63 expression level. They, after giving the radiation, they make radiation give a phosphorylation of a p63. However, cisplatin didn't make a phosphorylation. So that means they will give two different pathways to the oocyte data signal. However, those signals merge to the p63. And then also we found a really interesting thing that after giving the cisplatin signal, those apoptotic markers will turn on the oocyte. However, after making p63 knockout, they do not express any apoptotic markers. We do not know why, how p63 controls those apoptotic markers yet. And also, when we look at when we give a cyclophosphamide, gamma-286 signal will turn on. The top upstream marker is turned on. However, downstream of vaccine, C5 does not turn on. That means p63 controls the apoptotic pathway in the primordial oocyte. And then we also found that after giving the time, they turned on high level of OPA, mitochondrial fusion protein. They automatically turned on high level. And then we concluded that cyclophosphamide induces TAP63 phosphorylation and then gives signal to VEX, and VEX will control the OPA and mitochondria, and it relates to the cytochrome C release in the oocyte, and then will induce a apoptotic pathway in primordial oocyte. However, after making P6 knockout, cyclophosphamide did not induce the VEX activation, so they could not release our cyclone C, so they could not be apoptotic status. And then we concluded that no matter what, all those signals will give a different pathway. Oocyte has a P6 molecule, and then they will control the oocyte fate. So we can think about P6 is the main molecule to controlling, and at the same time we can prevent, we can kill the tumor cell. So I am thinking of those systems right now. How about immunotherapy on the ovarian functions? I wanted people to think about new cancer therapy. So immunotherapy is a new frontier. It's a revolutionized therapy, and then immunotherapy helps the immune system to better aid against the cancer. There are so many different kinds of immunotherapy. So currently, multiple immunotherapy were approved by FDA for childhood cancer treatment options. And however, they have so many side effects, including rash, diarrhea, and fatigue, and also sometimes the life-threatening toxicity by immune-related adverse events, something like diabetes, hepatitis, myocarditis, and nephritis, etc. And the myocarditis is really severe condition. However, we do not know anything about the side effect in reproductive function. So especially female, male, we do not know. And then I look at the Google PubMed, it's not reported many things. And then also, if you look at this recent review paper, mentioned the pembrolizumab and the nivolumab, and then there's no any report about this immunotherapy reagent. And then, so my lab is interested in pembrolizumab because pembrolizumab is a humanized mouse monoclonal immunoglobulin G antibody directed against the human cell surface receptor PD-1. And then in February 2019, pembrolizumab received its first approval in the adjuvant setting in the US for the patient with resected stage 3 melanoma. And then if you look at here, the PD-1 mechanism is that tumor cell expresses PD-L1, and then T cell expresses PD-1. And then they will bind to each other, and then tumor cell cannot be recognized as T cell. However, we give it the pembrolizumab. Pembrolizumab is red color. So red color bind to the PD-1 receptor in the T cell, and then they will, T cell can activated by the pembrolizumab. So cancer cell can be recognized as activated T cell. So, and then in 2020, 2017, one paper just came out. They wanted to figure out how pamperism maintained inside the body. They they labeled the pamperism, and then they traced the 168 hours, and then found that after 168 hours later, most of organ lost those isotope signal. However, ovary contains, if you look at here, ovary contains so much pembrolizumab. They didn't mention in the paper. However, I wondered what's going on in this ovary and how pembrolizumab affected the ovary. So my lab wanted to study quickly to see the side effect of pembrolizumab in primordial follicle. Although in patients, usually they get pembrolizumab every 3 weeks for over 1 year. So, but however, we just only tested we wanted to know the side effect of pembrolizumab, so we only injected twice. Especially we use the PD6 mouse because they have so many primordial follicles. And then first time when we look at histology, we couldn't see the difference between saline or pembrolizumab group. However, when we count the follicle numbers, we could see little bit, not a little bit, it's a little bit, almost a half of a percentile of primary follicle decreased with only twice of injection of pembrolizumab. And then we could see so many dying granular cell in antrum follicles. And also we wanted to see what's going on in this mouse after injecting of pembrolizumab, and then we wanted to see T cell inside the ovary, and we found that increased TCell3 positive cells inside the ovary after injecting of pembrolizumab. If we look at here, the top panel is a lymph node as a positive control, and then we wanted to when you look at the ovary, we could see the CD3 positive cell inside the follicle, between the follicle as well. But however, we do not know what they do inside the ovary, inside the follicle. And when we look at the surface area, especially there are a lot of primary follicles just in the surface area. They contain a lot of CD3 positive T cells in the ovaries. And also we found that so many inflammation happened, so showing that COX-2 and then TNF-alpha increase inside the ovary. And then most interesting, I found that ovary also expressed PD-1 and the PD-L1 protein. So PD-1 is expressed in surface area, and PD-L1 is highly expressed in oocyte cytoplasm. However, we do not know what they do inside the ovary. And also, when we look at the PubMed, everybody talking about PD-1 is a positive marker for ovarian cancer, not ovary and follicle, ovaries. So I'm really interested about this molecule. Maybe they have a special function, or we should advocate. We should mention to the oncology that we cannot just use pembrolizumab just killing the ovarian cancer. We should think about ovary as well. So, conclusion is the immunotherapy reagent. After using immunotherapy reagent, we could see ovarian reserve loss and the granular cell death, and the CD3 positive T cell increase, and inflammation also increased. However, immunotherapy is totally different from chemo or radiation therapy. So immunotherapy is TAP6 independent cell death, inducing cell death, and then also it's not immunotherapy. It's not induce specific pathway, and also maybe I do not know yet. Maybe we do not know yet, but so currently I can say that immunotherapy is not induce a apoptotic pathway. Also, immunotherapy is not give not acute signal into the ovary to decreasing primary follicles. So immunotherapy affects ovarian health with unknown mechanism. We should investigate how to protect ovarian health from cancer therapies, including immunotherapies for cancer patients. So the goal for the research women's health, we wanted to develop really applicable photoprotective reagent to protect ovarian germ cell. At the same time, should not compromise cancer therapy, and then eventually want to say maintaining women's systemic health. So in conclusion, for developing and analyzing photoprotective reagent, we also think about the radiation, chemotherapy, and immunotherapy as well. And at the same time, we should think about oocyte and granular cell, thicker cell, somatic cell, and environment of the ovary, their health, quality, quantity, and functionality. Also, we should think about hormone secretion, reproductive cyclicity, female systemic health, and then we should think about the next generation, if they are healthy or not, and then also their reproductive health as well. And also, I think we should not think about ovary. We should think about cancer itself, how cancer cell affect the ovary itself. So until the finding the photoprotective reagent, we will keep moving forward, and then I would be really happy when we find the really applicable photoprotective reagent. So thank you so much for listening. This is my precious lab members, Dr. E. Luan, she contributed a lot of study, and then Dr. So-Youn Kim, Ibrahim Irish, Rosemary Dong, Amir, Abajari Kia, and then Macy Tonin, and then my collaborator, UNMC, Dr. John Davis, Dr. Roy, and Dr. Coulter, they supported me to this study, and they always gave me this really great advice. And then my collaborator, Dr. Jing Zhu, and then my funding. And then this is Nebraska Medical Center. I think most people might not know where Nebraska is in Omaha, so I just put our Omaha and Nebraska Center is over there. Thank you very much, and I will happy to take any questions. Thank you.
Thank you for that really exciting talk. Can I ask a really silly question? I'm a clinician. So with the P63-mediated apoptosis, is that the same mechanism for apoptosis in ovaries in women who haven't had chemo?
Pardon me, could you speak a little bit loud?
So I'm wondering if the P63-mediated apoptosis from chemotherapy, is that a similar mechanism for the apoptosis when you're not receiving chemotherapy and, follicle death, in natural follicle death?
So you mean oocyte to die through P63 between the chemo-induced versus natural-induced follicle death?
Yes.
Yes, we do not know in natural oocyte death. However, it's no one knows yet. But chemo responded to P63 phosphorylation, which really occurred inside ovary. However, in natural oocyte death, people still think that maybe it's not a apoptotic pathway. It's not P63 related. So that's currently reported. That's what I understood so far. Thank you.
Thank you.
Hi, can you comment on proton radiation's effect on the uterus with respect to the safety of women carrying a pregnancy in the future after exposure?
Okay, thank you. So proton therapy is just an experimental stage right now. At the time, we have a collaborator was working collecting the human sample after treating proton. But we didn't analyze it yet, so I cannot say anything right now. But maybe in the future we can see the result after using proton therapy. Yes, I hope we can get positive result.
Okay, thank you.
Thank you.
Nice talk, So-Youn. So I'm so happy you got the consistent doxorubicin treated data. So we recently performed the sequencing, and actually the the most upregulated gene is the P21 after doxorubicin treatment. That is very important cell cycle regulator genes. So I don't know the answer, but I wonder whether you know whether those for example, P21 or other cell cycle regulator, they are the target of the P63 or P53 in the oocyte of primordial follicles.
Thank you for a great question. I do not know anything the downstream yet. So currently we wanted to know what P63 regulated what kind of gene regulate by P63. So I'm really right now working on with the knockout mouse model and then just the wild type mouse model with the treatment of cyclophosphamide or radiation. So I may give you answer later, some time point. Thank you.
Thank you for this very important work, Leslie Appiah, University of Colorado. So for the P63 knockout mice who received cyclophosphamide, there was no apoptosis, correct?
Yes.
Are we worried about any propagation of DNA damage from that cyclophosphamide, or were there repair mechanisms that corrected for that?
Thank you for a great question. So when we look at, we could see gamma H2AX signal after giving cyclophosphamide in P63 knockout oocyte. However, it disappeared over time. So we do not know DNA repair system occurred or initiated by certain molecule. But the recent paper from Dr. Volker from Germany, they do RNA sequencing, and then they found that DNA repair system is a total separate mechanism from the P63, but we do not know yet. Also, we do not know how primordial follicle respond so quickly by DNA damage. So this kind of things also for future research would be. Thank you very much.
So just amazing work. I think clearly everyone's interested in things that can be preventative. I just wanted to clarify, because the doxorubicin was found to have, through that mechanism, ovarian follicular death?
Yes.
Yeah. So, because I think also as a clinician, so totally from that focus, doxorubicin doesn't appear to be an agent that is implicated in ultimate, effect on the ovaries as it relates to fertility. So I'm just curious how that, how you understand, how that's understood in terms of maybe different pathways, or because doxorubicin is not a risk factor in large clinical trial studies for ultimate impaired ovarian reserve.
Yes. Thank you. Thank you very much for bringing up the doxorubicin issue. So we also wanted to know why we tried multiple doses of doxorubicin because doxorubicin is not the gonadotoxic enough, like cyclophosphamide or radiation or cisplatin. So, but however, when we increased the doxorubicin, as Dr. Shuao tested, it's a really high condition still kill the primordial follicles. So I don't know, it's not clinically applicable. But however, I think still, if doxorubicin kill the primordial follicle with a high concentration, how oocyte would die? I wanted to know the kind of molecular level study, and then what kind of topoisomerase inhibitor can kill the oocyte? Because the oocyte, I do not know well, but I don't think a primordial oocyte really need at the moment. They need topoisomerase. However, with the doxorubicin still kill the primordial follicle. So that's why I just tested it, although it's not the clinically relevant. Yeah, that's my answer right now. I hope it can be answered for you. Thank you. Thank you very much.
Again.